This talk is about the topographic histology of the central nervous system. Please, before viewing this video, see a prior video titled Overview of the Nervous System Histology. Here are the class expected outcome. You do not need to read them. We will use them to chart our talk. This talk will be conducted using a question and answer format. The first question is, please look at this specimen and choose which letter corresponds to the pia. This is a representation of the scalp, skull, meninges, and brain. A way to remember the layers above the skull is to remember the word scalp. The S stands for skin, the C for connective tissue, A for aponeurosis, L for loose alveolar tissue, and P for periosteum. Now I am indicating to an aponeurotic vein, bone, diploic vein, emissary vein, the epidural space. This space in the cranium is a potential space between the cranial bone and the periosteum that covers the inner surface of the cranium. Other names used to refer to the periosteum at this location is bony dura, periosteal, or endosteal layer of the dura mater. This layer is of mesodermal origin. I am now indicating to the meningeal dura. The meningeal dura is also of mesodermal origin. Sagittal sinus, venous lacuna, subdural space, which is also a potential space, arachnoid trabeculations bathed in cerebrospinal fluid, arachnoid granulations, artery, pia mater represented in red, gray matter, and white matter. I will now reduce the size of this diagram and place it in a corner, as you can see in this frame. I have done so to indicate a portion of the tissue in order to show its histological appearance. You can see the subdural space, the arachnoid, subarachnoid space, which extend from the inner surface of the arachnoid to the pia, and the gray matter. So the answer to this question is D. Next question. Please take a look at this specimen and select the letter that corresponds to a protoplasmatic astrocyte. I have reintroduced the same cartoon we used for the last question and indicated by a blue rectangle the area of interest, which I have enlarged. I have now indicated the white matter, gray matter, and the sulcus region. Let's first pay attention to the sulcal area. As you can see, I have placed the specimen to the side to show the histology of the area. Here you can see the pia. Notice the arrow in the corner specimen. The pia adheres to the parenchyma, as you can see. The pia consists of flattened cells with long, equally flattened processes that closely follow all the surface features of the brain and spinal cord. The pia and arachnoid together constitute the leptomeninges. The leptomeninges derives from the neural crest. The space from the pia of one convolution to the pia of the neighboring convolution is part of the subarachnoid space, as indicated. I am now indicating the arachnoid. Again, notice the aqua arrow on the corner specimen here and here. Also notice the arachnoid trabeculations. Now that we have looked at the sulcal region, we will look at the gray matter in the area of the red square. Here I have placed the specimen to the side to show you the magnified specimen. I am now indicating to a neuron. They are large, mostly round, with a dense nucleus and surrounding pale cytoplasm. At times you see one or more projections from their bodies. As you can see here, these projections are axons or dendrites. I have now indicated two more neurons. I will now indicate the second type of cell in the central nervous system, the glia cells. They are smaller than neurons and 10 times more numerous. This is another glia cell. Here you have two and another two and a few more down in the corner. So we have looked at the gray matter and now we are going to look at a very interesting area, the junction between the white and the gray matter. I have now displaced the specimen to the corner and magnify the selected area. Here we can see a neuron and another one and a bunch of more down here and here. This structure is a vessel. I have now indicated a bunch of glia cells on the upper left hand side of the enlarged specimen and now a few on the opposite side. Most of the ones in the left upper corner are oligodendrocyte. They look like fried eggs. As you know, they make myelin and therefore are close to the axons. Whereas most of the glia on the lower right side corner of the specimen are protoplasmatic astrocytes. The choice of calling protoplasmatic astrocytes is primarily based on their presence in the gray matter close to the neurons. Protoplasmatic astrocytes are often found between vessels and neurons as indicated here. When you find similar cells in the white matter at this level of amplification using this stain, there is no way of knowing what type of glia cells they are, but the best bet is that they are fibrillary astrocytes. And if you see linear nucleus in the gray or white matter, they are likely to be microglia, the third type of glia cells in the central nervous system. Microglia are the cells targeted by HIV-1 virus in patients with AIDS. So the answer to this question is C. Next question. Please take a look at this specimen and choose the letter indicating the dentate granular cells. The hippocampal formation, if dissected in a particular way, looks like a horse sea monster. It is from this appearance that the name derives. This is a schematic representation of the hippocampal formation. The hippocampal formation is made of three parts: the hippocampus proper or horn of Ammon, the dentate gyrus, and the subiculum. I will start by naming the boundaries of the hippocampus. These boundaries are the temporal horn of the lateral ventricle. I am now indicating to the floor of the temporal horn of the lateral ventricle, the choroid plexus, ambient cistern, and hippocampal fissure. The fibers are complicated. At this magnification we can see the fimbria, which goes on to form the fornix, which connects the hippocampus with the mammillary bodies, the alveolus, which form the fimbria, and the perforant pathway. In order to see the different cells of the hippocampal formation, we will look at two different locations. We will first look at this area, which I have now enlarged. There you can see the pyramidal cells of the hippocampus. We can also look in this area, which I have enlarged, to show you the dentate granular cells. So the answer to this question is E. Next question. Which of the following is not a three-layer cortex? A. Cornu ammonis 1, B. Dentate cortex, C. Subiculum, D. Entorhinal cortex, E. Hippocampal cortex, F. Cornu ammonis 4. This is an enlargement of the previous specimen. In it, we can see the entorhinal cortex, called so because it was originally believed to be inside the nose. We can also see the subiculum or support. Then we can see the hippocampus proper or horn of Ammon, which I have labeled here. This cornus ammonis has been divided in 4 parts, starting with the segment closest to the floor of the temporal horn of the lateral ventricle, CA1, CA2, CA3, and CA4. Now I have delineated using a red dashed line the dentate. The 3 components of the hippocampal formation are made of 3-layer cortex. Layer 2 of the hippocampus proper is made of pyramidal cells. Layer 2 of the dentate gyrus is made of granular cells. The entorhinal cortex is made of 6 layers. Between the 3-layer hippocampal formation and the 6-layer entorhinal cortex, there is a transitional area of 4 and 5-layer cortex. This is a coronal inversion MRI going through the hippocampus. This is the normal hippocampus. This is abnormal. This appearance is suggestive of hippocampal sclerosis. Mesial temporal sclerosis is the most common cause of temporal lobe seizures. So the answer to this question is D. Next question. Please look at these specimens and choose the one corresponding to a pyramidal cell. The pyramidal cells are usually easily recognized by their pyramidal shape, bed cells being the largest. Purkinje cells are also recognized by their shape, but also by their position between the dense granular cells and the relative devoid of cell neuropile called the molecular layer. The Cajal-Retzius cell is an odd cell. It looks like a pyramidal cell out of place, near the surface of the cortex. They are the project manager of the cerebral cortex formation, and therefore they are rarely found in adult or children. They are not involved with information processing. So the answer to this question is A. Next question. Please look at these specimens and choose the one corresponding to the caudate nucleus. Specimen A main feature is diffusely spread non-pyramidal cells. The neuropile has the usual characteristic consisting of dense network of interwoven nerve fibers and synapses together with glia filaments. Cells are non-pyramidal neuro and glia cells, as you can see in this frame. This specimen can be found in any basoganglia. Specimen B main feature is also diffusely spread non-pyramidal cells, as you can see here. The neuropile has the same characteristics as the previous specimens, but at slightly higher power they seem a little bit larger. But the defining feature is an interface with CSF covered by ependymal cells. Notice the little hair projecting into the CSF. This specimen corresponds to the caudate nucleus, since it is the only basoganglionic nuclei bathed by CSF. The C specimens is characterized by columnar bands or rows of pyramidal neurons, as indicated here. The neuropile has no specific characteristics. Arachnoid layer is present. This appearance is typical of cerebral cortex. Specimen D consists of two clearly demarcated areas by a fence of Purkinje cells. Towards the Purkinje cells dendrites, we have regular neuropile, the molecular layer. Towards the Purkinje's axon, there are so many small cells that the neuropile can hardly be seen. This is called the granular layer. We can also see the arachnoid. This specimen corresponds to cerebellar cortex. So the answer to this question is B. Next question. Please look at these specimens and choose the one corresponding to the visual cortex. Despite difference in size and different stain, the structure labeled A can be easily recognized as the hippocampus. The next structure is perhaps less familiar to you, and I will spend a few minutes explaining it. This drawing depicts the origin of the indusium crisium. Let us first direct our attention to the new schematic representation of the brain. The hippocampus formation is a massive structure connected to the mammillary bodies by the fornix, traveling under the corpus callosum. The brainstem and the cerebellum are behind. In the rat, the hippocampus formation migrates in a rotatory fashion. The result of this rotation and the downward displacement of the mammillary bodies leads to arching of the fornix and thinning of the anterior-most part of the hippocampus, which, as it was in the nude, is on top of the corpus callosum. In humans, the process continues with further arching of the fornix and further thinning of the anterior-most part of the hippocampal formation above the corpus callosum, ending up like a thin strip of gray matter at each side of the corpus callosum, as indicated here. Next, we will look at the cortices of the two specimens presented in this question. This is a parasitical cut of the brain. You can see the enclosed area in the blue square, which is a portion of the cingulate gyrus, and now a portion of the calcarine cortex. I have now enlarged both areas. Notice the thick dark band of myelinated fibers. This band is called the band of Gennari and consists of a prominent outer band of Beilanger. As you can see here, the stria or band of Gennari can be seen in a fresh specimen as a white line. So the answer to this question is D. Next question. What is the name of the structure indicated by the curved arrow? A. Virtual Robin spaces. B. Subarachnoid spaces. C. Subpial spaces. D. None of the above. I will use this cartoon to explain the virtual Robin spaces. This is the representation of an artery penetrating the cortical brain parenchyma. Now I want to call your attention to the pia. Notice that at this point the pia divides. One division continues with the artery into the brain parenchyma and then fuses with the wall of the vessel. The other division cuffs the artery. Above the pia we find the subarachnoid space represented in light blue. Below we find the neuropile represented in pale pink. The edge of the brain parenchyma is lined by the feet of astrocytes, as indicated by the multiple arrows. The space between the pia and the vessel wall, as indicated by the arrows in this frame, and deeper into the parenchyma, the space between the astrocytic feet and the structure formed by the fused pia mater and vessel wall, as indicated by the arrows in this frame, is the virtual Robin space. This arrangement is seen in cortical arterioles entering the cerebral cortex. Veins and arteries penetrating the basoganglia have slight different blood-brain barrier arrangements. Hence, despite the difference alluded to, the virtual Robin spaces, regardless of being around arteries, arterioles, veins, and venules, do not communicate with the subarachnoid space, and the fluid inside the virtual Robin spaces is not cerebrospinal fluid. The fluid inside the virtual Robin spaces has more similarities to interstitial fluid. There is evidence to suggest that virtual Robin spaces are part of the lymphatic drainage pathways of the brain. So the answer to this question is A. Next question. What is the name of the structure indicated by the arrows? A. Virtual Robin spaces. A. Area postrema. C. Neurohypophysis. D. Organum vasculosum lamina terminalis. A good place to start answering this question is the blood-brain barrier. The blood-brain barrier is formed by endothelial cells with tight junctions and no intercellular cleft or fenestrations, thick basement membrane, and astrocyte food processes surrounding the thick basement membrane. Pericytes are cells present in the intervals along the walls of capillaries and postcapillary venules. In the CNS, they are important for blood vessel formation, maintenance of blood-brain barrier, regulation of immune cell entry to the central nervous system, and according to some authors, they are also important for the control of brain blood flow. They are not considered part of the blood-brain barrier themselves. The circumventricular organs in the brain are small, highly vascularized midline structures located in the walls of the ventricular system. Circumventricular organs lack blood-brain barrier, except for one of them called subcommissural organ, which I will mention later. Circumventricular organs have been proposed to have a role in many homeostatic and non-homeostatic functions. Circumventricular organs are all single and midline except for the area postrema, another of the circumventricular organs that I will mention later. This circumventricular organ, that is the area postrema, is close to the midline but bilateral. Circumventricular organs can be divided into secretory and sensory organs. I will now proceed to mention them. Most authors talk about 7 circumventricular organs. 1. The medial eminence organ is a depot for hypothalamic hormones. It has a secretory function. It is located in the medial eminence, which is located anterior and medial to the mammillary bodies. 2. The pineal gland also has a secretory function. The pineal gland releases melatonin and it is part of the system controlling sleep. 3. The mysterious subcommissural organ is believed to have a secretory function related to the formation of the posterior regions of the diencephalon. The mysterious subcommissural organ, which unlike all other circumventricular organs lack arterial fenestration, is a common question in board examinations. 4. The neurohypophysis organ has a secretory function. The neurohypophysis organ stores and releases oxytocin and vasopressin. 5. The vascular organ of lamina terminalis has a sensory function. The vascular organ of lamina terminalis is sensitive and osmotic pressure of blood. 6. The subfornical organ has a sensory function. The subfornical organ is sensitive to a wide variety of hormones and neurotransmitters. And the 7th, the area postrema, indicated here. The area postrema is located on the dorsal surface of the medulla oblongata and the caudal end of the fourth ventricle. The area postrema is connected to the nucleus of the solitary tract and the dorsal motor nucleus of the vagus, making up the dorsal vagal complex. The area postrema has a sensory function. The area postrema is considered the chemoreceptor trigger zone for vomiting. As you can see, it's a bilateral organ. So the answer to this question is B. Next question. Where is this type of neuron usually found? A. Parasympathetic ganglia. B. Sympathetic ganglia. C. Spinal dorsal root ganglia. D. Basal ganglia. Based on the number of neurites, neurons are classified as multipolar when they have more than 2 neurites emerging from the pericardium, bipolar when they have 2 neurites coming out of the pericardium, and unipolar when only 1 neurite emanates from the pericardium. Unipolar neurons are usually called pseudo-unipolar because embryologically they originate as bipolar neurons and subsequently become unipolar, as portrayed in this illustration. At the center of the specimen I have just introduced, you can see the body of an unipolar neuron, its single emerging process, and the split. I have introduced this cartoon to emphasize that the neurites on both sides are axons. Unipolar neurons are sensory neurons with cell bodies located in the spinal root dorsal ganglia, as indicated here, and in the cranial nerve sensory ganglia, as you can see here in the trigeminal ganglion. I almost forget to tell you that dorsal root ganglia and cranial nerve sensory ganglia also house visceral sensory neurons. So the answer to this question is C. Next question. Where is this cell likely to be found? Please take a look at these images and select the correct one. Despite its appearance, this cell is a pseudo-unipolar neuron. Notice the central position of the nucleus and nucleolus. This configuration is a very characteristic feature of pseudo-unipolar cells. In this added structure, I am indicating to one unipolar neuron. And in this now added specimen, you can appreciate that we're dealing with a dorsal root ganglion. So the answer to this question is D. Next question. Where is this cell likely to be found? Please take a look at these images and select the correct one. The cells in this question is a multipolar neuron. This is often hard to say based on the appearance of the multipolar neuron on many specimens. Multipolar neurons are probably the most frequent type of neurons encountered in the nervous system. I will now show you some examples. This is the classic multipolar neuron. It is a pyramidal cell with its thin axon and multiple beaded dendrites, reflecting the multiple synaptic terminals going on to them. These cells is found in the cerebral cortex, especially prominent in the external and internal pyramidal cell layer. This is a cortex specimen showing multiple pyramidal neurons. I have now introduced the largest pyramidal neuron, the Betz cells. These cells are present in the primary motor cortex. Multipolar cells are also found in the parasympathetic and sympathetic ganglia. These ganglia house the motor neurons of the peripheral autonomic system. This specimen was taken from a sympathetic ganglion. In this enlargement, you can see the characteristics of these cells, their eccentric nucleus and nucleolus. Multipolar neurons are also present in the spinal cord. In this spinal cord specimen, we can see white matter surrounding an anterior horn. The area I have enclosed and now enlarged shows a vessel and multiple clusters of cells, as you can see here and here. I have now migrated this specimen to a new frame, selected an area, and enlarged it. Here you can see an undeniably multipolar neuron. This one is more difficult to tell. This one, despite being multipolar, looks bipolar. This one looks multipolar, and this one looks to me like a little fish. Yet still is a multipolar neuron. I have again migrated the specimen, selected and enlarged the area of interest for you to better see two neurons in it. They are both lower motor multipolar neurons despite their different appearance. Remember that we're looking at three-dimensional structures in two dimensions, hence distorting their appearance. And also remember that neurites are very difficult to stain. It is important to know that not all multipolar neurons are motor neurons. Multipolar neurons, as you can see in this cerebellar specimen, include Purkinje cells and granule cells. So the answer to this question is A. Next question. Where is this type of cells likely to be found? Please take a look at the different images and choose the correct answer. The cell in this question is a bipolar cell. They usually have a dendrite and an axon. The axon has collateral and terminal branches, as you can see in this frame. These are two examples of bipolar neurons. The body of the cell is ovoid. You see a process on one side and another one in the other side. This is another example. You can see the pericardium, one process going up and another one going down. Here you can better appreciate the process going down in a different neuron. Where do we find bipolar neurons? Bipolar neurons can be seen deep in the cerebral cortex, as indicated here. These neurons are very rare and only found in human and ape. They are called phoneconemon neurons. Bipolar neurons are also found in the retina. I have selected, enlarged, and labeled the different retinal layers. At this junction, you do not need to memorize them, but you do need to know that bipolar neurons are present in the inner nuclear layer of the retina. Bipolar cells are also present in the nasal neuroepithelium. They are the olfactory receptor cells. Their centrally located axons form the real olfactory nerves. Bipolar cells are also present in the neurosensory organs of the inner ear. Here I have selected and enlarged an area of the spiral ganglion, so we can see the bipolar cells whose axon will form the cochlear nerve. The other internal ear organ with bipolar neurons is the ganglion of Scarpa, which I have selected and enlarged so we can see the bipolar neurons that will produce the vestibular nerve. So the answer to this question is C. Thank you very much for your attention.